your host, Dennis Horvitz, the David Letterman of the Hopelessly Damned. We are doing another one of our Sunday brunches. Before I forget, if you would like to know more about our Sunday brunches or anything else that's happening at New York City Atheists, you can contact us at NYCAtheists@gmail.com, and we can put you on the our newsletter email list. In any case, we are taping this on Mother's Day, and I can't think of a more apt subject to discuss than the human genome, because if we didn't have mothers, we wouldn't have a genome. Lame segue, but I'm like working under pressure. What exactly is the human genome, and why is it important to know about it? Well, because someday that knowledge may save your life, because it may determine actually how long you live, and it will completely change the way medicine is practiced. Our guest is Dr. W. Richard McCombie. He is professor at the world-renowned Cold Spring Harbor Laboratory in New York, Cold Spring, New York. It's also the home of 7 Nobel Prize winners. Dr. McCombie has spent 4 years at working on sequencing the human genome at the National Institute of Health in Bethesda, Maryland, and he spent all the time since then, I guess starting in 1992, he has been working at Cold Spring. So without further ado, Dr. McCombie.
A disease is caused by a single change in one of those 3 billion bases. Not always, but very often. There's a number of genes. There's a number of diseases where that's the case. If you think about it, finding one of those changes is a lot easier if you actually have the books ahead of time. Not easy, but easier than if you think there's 3 billion possible places where this change could occur and I don't know any of them or don't know anything about it. So one of the reasons for doing the Genome Project was in fact to get that reference telephone book, the book from one person to help find any of the genes that people were looking for diseases. And that was one of the major purposes of doing it. And politically, the disease that really drove this through funding in Congress was cancer, because of the difficulties in finding what was known to be many genes involved in cancer, some of them known by the time the project started, but it was clear there would be a lot of genes involved and they would be interacting, and having this phone book would be invaluable in understanding what all those components were and the interactions. So the first version of our genome, the human genome, the project was started, depending again on when you consider it, in around 1990. And the first version was published in 2001, in February 2001. And this is the cover of the issue of Nature, the scientific journal, British scientific journal actually, where this was published. And this was in fact a collaboration of probably in the order of 1,000 people. My lab was one of the smaller labs. There were 20 labs that participated in the project, so I think we were the 19th largest. The largest one was actually in the United Kingdom, the Sanger Institute, and they probably sequenced almost 1 billion of the 3 billion bases. Now, when I say this is a reference sequence, and I'll come to this later, each of us have differences between our genomes. This is a sequence that's about 70% from one unknown individual from upstate New York, and the other 30% is from about 7 or 8 individuals, most if not all of them anonymous as well. And this was something that happened early on, where they realized there's so much information in here about that individual that they wanted it to be anonymous. The downside is, if we find something interesting in here, we have no idea what they're like. So if we find that this sequence has something that maybe put them at high risk for cancer, we have no idea who it's from, so we have no idea if they are actually have gotten cancer, for instance. So there's trade-offs there between the ethics and the privacy concerns and the scientific concerns, and that's actually been an overriding issue that we're still grappling with as this accelerates, as I'll talk about in a bit. So how will this affect how we do research? And I think I mentioned this before, but it used to take literally weeks to years before genomics to identify some of the genes. when we first started doing this, when I was at NIH, I got an email from someone, it was early days of email even, and he said, I've been trying to clone a gene for a neurotransmitter receptor involved in neural transmission from this organism that we were sequencing for 10 years, and I haven't been able to do it. I noticed it in the database. How did you do it? And I actually said, we did it by random. We basically, the idea is sequence everything. And because the technology had so changed that instead of people spending weeks or months or even years to refine what they sequenced, we literally plated out things on a plate, picked everything that grew and sequenced it because the technology had changed so much. That was sequencing at a level, as I'll show later, many orders of magnitude below what we do now. So what do we see when we look at the genome? First of all, since this is a reference genome again, and I'll talk about individual genomes in the second part of the talk, we get a much better understanding of the same is in fact true in plants. So this is a diagram. It's called a crop circle in the trade as a joke, but it in fact shows a diagram of the rice genome, the corn genome, and the wheat genome. This is 435 million bases. This is about 2.5 billion bases. This is 16 billion bases. But if you look at it, this is basically, and these are all cereals, not all plants are like this, but all the cereals. Wheat and barley would be in between here, between corn and rice. What you see is basically the rice genome. The essential components are largely in order, but spread out, and in fact duplicated in the corn, and then duplicated again in the wheat genome. But that is the footprint of plant evolution. And very important genes like this gene, which is actually, I believe, the height gene, one of the main genes involved in the green revolution of the 60s that led to higher yielding plants, is in a same relative position in all three genomes, as well as barley and wheat. Perhaps nowhere is the footprint of evolution more apparent than in olfactory receptors. Olfactory receptors are the genes that are the signaling molecules that let us smell things. So when you smell something, we had our comparative genomics and evolution, and it's very clear from looking at these genomes how evolution has occurred and many of the changes that have happened during the evolutionary process. We also get a better understanding of the range, especially as we do more and more individuals, of the variation and how that affects the phenotype. So the term phenotype is a term biologists use to describe sort of what you're like. So blue eyes would be a phenotype. And so it's susceptibility to cancer, for instance, and the things that biologists are more interested in, blue eyes, but they're all phenotypes. So comparative genomics are our bunch today. There are these receptors in your nostrils, chemicals in the food bind to them and send a signal. They're very specific for certain types of odorant molecules. And the reason we can smell lots of different things with some nuance is because we have many, many of these receptors in our nostrils. It's also the fact that they vary a little bit from one person to another. It's why we sometimes may really like the smell of something like licorice makes me gag. My wife loves it. It's probably different in some of those receptors. But the interesting thing here is, again, this footprint of evolution. The mouse has roughly 1,391 of these genes. We have about 800. So we've lost some of these. Even more important, though, in looking at this as the footprint of evolution is, and this is not work from my lab, I should point out, this is a number of people as shown down here. There's something that happens called the development of a pseudogene. And what a pseudogene is a gene, again, the gene is the thing that makes a protein in our body, that has a change in it, a mutation, if you will, that makes it no longer functional. And we can tell that most of the time on the computer. And when we look at these olfactory receptors, what we see is that in humans, over 50% of the 800 genes are actually functionally inactive. They can't work. So we've lost more than half of the genes that the mouse has. So these are all, roughly these 802 are all in the mouse. We've lost a few that are totally unrecognizable and we don't see. But even of the ones that we can look at and say, oh, that's an olfactory receptor, more than half of them no longer function because they've gained mutations during evolution. But the residue is still there. So this is a case where we have hundreds of genes which don't function. The only reason we have them is our common ancestor with the mouse had them, because they're not functional entities. Even more interesting is if you look at primates. And again, about 25% of the mouse olfactory receptor genes were inactive, and about 50% of ours, chimpanzees and so forth, are in between. Which, if you think of the biology of these organisms, how much a mouse depends on smell versus a chimpanzee versus us, makes perfect sense. We are not nearly as dependent upon our sense of smell for things like running from prey as a mouse is, or even a chimpanzee. And so this is consistent with the biology of the organism. And again, the chimpanzees have roughly the same number of 800 to 1200, with about a third of them inactivated because of mutations. But the residues are still there, showing that their common ancestor actually had these, as did ours and the mouse common ancestor. They just no longer have them as part of their repertoire for smell. So for the second half, and really, well, a bit more than half, I want to talk about individual genetic variation. Again, remember that first genome sequence took about 10 years to do. It took 600, 800 people, minimally. I've seen the cost estimated anywhere from 50 million to 300 million. I actually was describing this process, giving a tour to our lab to former Governor Spitzer, actually, and I said it took 10 years, and he said, really? I would have said it took 100,000 years. He said, well, fair enough point, because it took us that long before we could do it the first time. It took us 10 years once we started trying. But that took anywhere from 10 to 100,000 years. So I want to talk about why individual variation, not just, again, this reference composite sequence that we did over a 10-year or 100,000-year period, but about individual sequence. So the reason that's important is everyone in this room here on each of our chromosomes has about, on average, one variation from the next person per 1,000 bases. So if you think of that, over 3,000 bases, that's about 1 million variants between any two individuals. Those variants are really, at a biological level, responsible with interaction with the environment, but are responsible for our individuality, our susceptibility to disease, our susceptibility to environmental insults, our longevity, as well as, to some extent, our behavior. This is an interesting example that I wanted to use just as an example. So this shows actually a genetic change between individuals, and it's going to be a little hard to see. This was a gel analysis done by someone in our lab. This is actually a change involving more than one single nucleotide, and so I talk mostly about variation in terms of a single change out of that 3 billion. This is a change that involves a segment of about 100 nucleotides that are contiguous. What you can see here is that this same region, without going into details, is present in 3 different forms. A large form, a medium form, and a small form. And I apologize, that's hard to see. Any individual is going to have 2 of those. One from each chromosome, one from their mother, one from their father. They can have 2 of the same. They can have two medium, like this person, they can have a medium and a large, they can have a medium and a small. They're independent events, but they have 2 of these. What this paper shows is the impact of these. And what these researchers found is that depending on which version of these you had and your environment, it had a statistically significant impact on your behavior. One form of this region of the genome, if you were abused as a child, makes you much more likely to have criminal behavior or abusive behavior towards your own children than if you have the other two forms. If you're not abused as a child, having that form has no impact on your behavior statistically. Again, this is statistical. It's not an individual situation. But if you take 1,000 people that have one form and they're abused as children, they're much more likely to be abusing parents than 1,000 people who have the other form. So people who have the other two forms, it doesn't matter if they're abused or not, they're very unlikely to be violent or abusive as adults. People who have the abusive, the at-risk form, if you will, who are not abused have no more likelihood to be abusive as adults. It's only the combination of the genetic background and the environment that lead to the phenotype. I use this example when I teach sometimes, and one student actually raised her hand. She said, "That makes me very uncomfortable." And my response was, "Good, that's why I showed it to you." Because this is more and more, this is first example of this, maybe it's in 2002, a year after the genome was completed. As you'll see, these are going to become more and more and more common. So, the future of this field. There are very recently, and by that I mean the last 3 years maybe, 3 to 4 years, been incredibly profound changes in the way we can do this. It's been a little longer than 18 months now, although it's still actually changing at a very rapid rate. They're going to completely change our ability to understand individual variation of the genome, because they're going to let us capture that individual variation and study it in a way that I would have never thought possible at the time the first reference sequence of the genome was done. This shows our lab now. It's actually a couple years old, and there's these machines in there. I just want to show this because I want to show a little bit of how this is done, because it's really quite amazing technology that was developed by a couple different companies. We use one from a company called Illumina, originally developed by a company called Solexa, and England that Illumina bought. Not really relevant to the scientific issues. Each of these boxes now, so I told you it took about 10 years to sequence the first genome, about 600 people. One of these boxes will sequence the complete genome in 6 weeks now. And I'll show some more examples of how those numbers are changing. The way these work is actually quite fascinating. They take actually individual DNA molecules and they put them on a solid surface the size of a microscope slide. Millions of them, hundreds of millions of them. They then add the 4 bases of the genome, A, C, G, T, each with a different colored dye on it. And they add them in a way by chemical modifications I don't want to go into, so that each of those molecules on the surface gets 1 molecule added to it. Then they take a photograph of these hundreds of millions. They actually take tens of thousands of photographs. So one of these machines kicks out about 8 terabytes of image files in a course of a run. Each of them showing the 4 different colors as each molecule, each of these bases is added. Now, as I said, that I hope I said that the genome is a chain of these ACTG in order. And so what this does is basically let you actually read how the bases are incorporated. So this molecule here, the sequence of the bases is TAAGT. And it does that just by taking sequential images after adding 1 base at a time and seeing what color gets added. It's that simple. And that's how it determines the sequence of these molecules. And it does it on hundreds of millions at a time. That's actually just a photograph of one of the base additions. And the bright ones you can see are all the molecules where a given base was added. You can see some darkness in the background. They're ones that didn't get that base added to them. So this technology is really completely changing the way we do genetics of human disease. It used to be, because it was so hard to sequence DNA, that we would look for indirect methods to find variants. So we would, for instance, look in a family and say, is there a big chunk of the genome that is inherited within this family that is associated with whatever disease we're looking at? And that initially, we would look for that even under a microscope, that big a change, which would involve tens of millions of bases perhaps. And then there become a lot of tricks that don't need to go into to look at more smaller areas, more refined areas, but still fairly large areas. But what we're really getting to now, because of this new technology, is being able to skip those indirect methods and looking for the best methods to do it more directly. We've always been limited by essentially cost, being time and money, and sort of the window that we would look at in the genome, because we didn't have the technology to look at the whole thing. And we have some number of patients we would need to look at in order to understand the difference between patients and non-patients. And if you multiply those together, that would give you some kind of cost, and that would really tell you if you could do the things. And I want to show an example of one we did in 1990 to show how that worked, and then one that was ongoing in our lab now. So this was a thing published in, from when I was at NIH, and what we looked at was we were looking for a Huntington's disease gene. We looked at 58,000 bases in one patient, because we were hoping we would find the Huntington's disease gene there. And we found 72 DNA variants in that 58 kilobases, 58,000 bases. That took about, well, I'll show, but it was a major effort for a big part of my life. Literally, I remember one day when I was asking myself, what am I doing? Because I encountered in the same day 2 morning rush hours. I worked from about 7 in the morning until 5 the next morning. And that was pretty typical. There were several of us working 100, 110 hours a week trying to do this. This shows the sequence output in my lab because of this new technology, and that was, again, 58,000 bases. This shows the sequence output with this new technology from August of 2007 to March of this year. You can't read the numbers very well, but the conditions, but what this number here is, which is about where we're at March of last year, is 1,400 billion bases, 1.4 trillion bases in a month, with a smaller group than that group that did 58,000 bases in 18 months or so. How that translates can also be seen here. I show a couple slides like this because it's really such a profound difference. The top slide is the number of bases generated by everyone in the world and put in GenBank, which is a NIH-operated public repository of DNA sequence. And you can see the growth from 1982 up to about 100 billion bases a year being put in GenBank. This is the same output by month in our lab. And you can see, and these are not exactly the same because this is raw sequence and this is a combination of raw and multiplexed, and it's a little different. But nonetheless, what you're seeing is a relatively medium-sized lab is doing more now in a month than everyone in the world combined did 10 years ago in a 5-year period. That kind of change has just an absolutely profound impact on the possibilities that we can do to find this individual variation in one person's genome compared to another. So one of the major projects we're doing is trying to use this technology and figure out how best to use it to understand the genetic changes that make people at risk for cognitive disorders, schizophrenia, bipolar disorder, and major recurrent depression, and ultimately with the hope of developing better disease diagnostics based on the genetic variants that are found in those individuals. And that's a big part of what we're using this new technology for. It's the biggest part. I want to show an example project of this to contrast to that project we did at NIH. This was a project started in 2007. It's not even at the level, with the technology where it is now, because the other thing, I actually want to go back to this because I didn't point this out. People have heard of Moore's Law. Moore's Law is the law from Gordon Moore at Intel about transistors per millimeter, I think it is, in integrated circuits. And it governs a lot of what happens in computers, and people use it as an example of our rapidly advancing technology. And Moore's Law states that it will increase by a certain amount over a certain period of time. If you plot Moore's Law on here, it comes out this period to about here. We're going between 2 and 4 times as fast as Moore's Law. That's actually causing problems for us because we where it's getting to the point where we can't buy fast enough computers. So this project is a gene that was previously identified in one family as probably causing schizophrenia by our collaborators in Scotland, and it's about 550,000 bases in like kbs kilobases, thousands of bases. As I said, the other one was about 58 kilobases, so this is about 10 times the size. That other project in 1990 we did in one patient. In this one, which is 10 times the size, we did in 1,000 patients and 1,000 controls, unaffected. And that's a pretty good indication of the change in technology. So again, this was roughly 10 to 20 people on that project I showed from 1990. 18 months to complete. Cost is about the same. We sequenced about 60,000 bases. Now with the technology, with roughly the same effort, actually probably fair to say not as much effort, we sequenced a composite of about 750 million bases. So a total change in the possibility of what we can do. It's accelerating even more. Again, we started that project in 2007. This is the approximate raw cost to sequence a human genome. So the reference genome, again, I used about 50 million here. The bigger the circle, the more the cost. And then the years go up. This is just order on the bottom. It doesn't really have a meaning. So the human genome reference was about 50 million. Craig Venture's genome was about 30 million. Jim Watson's was a few million. And it keeps going. And there's actually, although you can't see it, there's a dot up here. You can't see it because it's so small. So we've gone from 50 million minimum here. Some people have said it's more like 300 million. It's 50 million. It's now, at the beginning of this year, it was about 25,000. It's now about 9,500. There are two companies that are predicting it will be between 100 and 1,000 by the end of the year. I actually don't believe either of them. It probably will be, though, 1,000 by the end of next year.